A very good day to everyone. My name is Fan Chayen. Now is going to present on my bioinformatics project 2. So this is my research topic, deep learning method in the prediction of mRNA-based vaccines. So in this presentation, we'll include 6 chapters, which are the introductions, the literature review, research methodology, research design and implementations, result analysis discussions, and lastly is our conclusion. So first and foremost, chapter 1 introduction will include some problem background, objective, and research scope. So first introductions, the coronavirus disease 2019 pandemic is currently spreading sustainably and affecting more than 120 countries. So according to the research, the severe acute respiratory syndrome coronavirus 2 is the potential source that derive the COVID-19 disease. So both are the coronavirus disease carried by the pathogen virus as the physical host and they can transmit it quickly. As a result, the virus host pass through the respiratory system, which may lead to a serious failure of the lung functions and difficulty in respiration. So therefore, the development of vaccine are critically significant and it can act as the immune system to fight against to the virus. So after vaccination, the immune system can respond immediately to the target virus and break down them to prevent disease infection. So the problem background in this research. So currently the COVID-19 has overspread globally and there are approximately 4.55 million total cases and 35,745 death case in relations at the time of writing 25 June 2022. So therefore, the development of the COVID-19 vaccines have been offered with an urgency approved to entering up from this pandemic. However, the COVID-19 vaccination is still exposes multiple challenges and difficulty on their efficiency and their stability. So one of the bigger challenges is that mRNA-based vaccines is incredibly fragile and must be kept in the uninterrupted cold condition for the storage and transport purpose. So thus, the investigation and study on the reactivity of mRNA bases required to be continued to discuss in this research. So the research objective in this research. The first is to analyze the mRNA vaccines and the existing computational algorithm and technique based on the long short-term memory network method. Second is to propose a LSTM method to predict the reactivity of mRNA sequence. And lastly is to validate and verify the performance of the LSTM methods in terms of mean correct root mean square error, MCRMSE, in the prediction of the reactivity of mRNA sequence. So we continue with the chapter 2 literature review. And in this chapter, we'll include some brief introduction about the COVID-19 virus, variants, and vaccines, machine and deep learning toward the COVID-19, and the structure about the RNN and the LSTM network. So the first is the COVID-19 virus. And the COVID-19 is under Coronaviridae family with a cloud-like pattern. And the virus is built up by the 4 structure genes, which are the envelope, spike, membrane, and nucleocapsid. And since that the viral protein entering the target cells through the membrane receptor on the S protein, so therefore the S protein is the significant marker for the COVID-19 vaccine developed. So the most common symptoms for the COVID-19 are fever, cough, tiredness, and the loss of taste or smell. So lastly, how does the virus spread? So the first out is through the droplets. So this is the most common transmission. So when an infected person coughs, so the droplets will carry the virus into the air so that anyone who is within 1 meter of that person can breathe it into their lungs. So second is through the airborne transmissions. So the research show that the virus can live in the air for up to 3 hours. So that can get into your lung in someone who has it breathed out and you breathe that air in. So lastly is the surface transmission. So the virus can live on the surface like the plastic and stand still for 2 to 3 days. So when you touch the surface that someone who carry the virus, so that you may have the risk to get the infection. So the coronavirus infection is start with the specific binding of the S proteins to the cellular entry receptors. So the virus will express and replicate their genome RNA to generate a complete full length of copies and incorporate them into a brand new viral components. So however, there might be a few mistakes in the genetic arrangement during the replication processes. So this kind of the changes in the DNA sequence is known as the gene variants. So this because the mutation of the coronavirus might influence their behavior, which include the speed of transmission rate, the related disease seriousness, and the effectiveness of vaccine and so on. So this is the example of the variants of concern. So the first one is the alpha, who found at the UK on the May 2020. You can find that the alpha can spread more easily. So continue with the gamma and the beta. So the beta is found at the South Africa on August 2020, while the gamma found at the Brazil on November 2020. So both are found that can split more easily and some vaccine may be less effective against it. So continue with the delta found at the Indian on October 2020. It can split more easily. Symptoms may present differently, may reduce the vaccine efficiency and still protect against several disease. And lastly is the Omicron, found on multiple countries on November 2021. In the earlier studies show that it can spread more easily. So that the government have been conducted various forms of prevention for this severe global disease. And the most significant effort is the development of vaccines for COVID-19. So there are several technology platforms designed for COVID-19 vaccines. So the first one is the virus host. The whole virus is used after it has been killed with the heat or chemicals. So it is safe because the virus is already died and it's easy to make. So the disadvantage is not as effective, so sometimes will make the disease more worse. So the group testing involved the Sinovac. And the second type of vaccine is viral vectors. A harmless virus has been taken and used to deliver the viral genes to build the immunity. So the advantage is the live virus tend to elicit stronger immune response than the died viruses. And the disadvantage for this is an immune response to viral vector could make the vaccine less effective. And the group testing involved the University of Oxford and AstraZeneca. And lastly is the nucleic acid vaccines. So this vaccine is use the DNA or RNA molecule to teach the immune system to target key virus proteins. It is easy and quick design. It also safe to use because does not interact with the genome. And the one of the disadvantage is it is necessary to keep at the cooler temperature. And the group testing involved of Moderna and Pfizer. So to address these global COVID-19 vaccines, many professional scientists, chemists, and healthcare specialists has exposed new technologies to track and control the virus growth. So the greatest use of technology nowadays is artificial intelligence, which can track the speed and detect the growth rate of the viruses. The AI acts as a broad umbrella that consists of many sub-areas. So the machine learning and the deep learning are the subset of the AI areas that consist of several algorithms that provide intelligent models to identify and cluster the particular task. So that the machine learning consists of the algorithm modeling culture of statistical model, and it only needs a small amount of knowledge to learn how to solve the problem. So the example of the machine learning techniques are the logistic regression, random forest, and the KNN. So deep learning, on the other hand, is the subset of the machine learning that focuses on building the deep structural neural network model that learn from the data using algorithm of feed forward and back propagation. The deep learning algorithm usually involve the deep neural network and deep convolutional neural network. So the outgoing development in the machine learning and deep learning. So there are few successful examples of the method for vaccine discovery. So the first one is the CNN used for the binding predictions of the peptide, and continue with the SVM, random forest, and the recursive feature selections is used to identifying the antigen from the protein sequence. And lastly is the LSTM network, which is stimulated sequence of the S protein to identify the possible target for the vaccine design. So the RNN is the powerful and the robust type of the neural network that helpful in modeling sequence data. So because this is the only one with the internal memory, so with the memory in the hidden layer, so RNA can help to remember the important thing about the input they receive, which allow them to be more precise in predicting what is coming next. So that's why they are preferred algorithm for sequential data like DNA or RNA sequence. So however, there are two major obstacles in the RNA algorithm, which are the exporting gradient and the vanishing gradients. So fortunately, the deep learning algorithm as like the LSTM network can handle these kinds of issues. So a brief introduction about the architecture for the LSTM network. The LSTM can also define as the memory block which responsible to store and organize the information by help of four main of components, which are the first one is the input gate, which is responsible to controlling the flow of information into the cell state. And the forget gate is identify the information from the previous cell, either keep or remove the information. So the third is the cell state, which is responsible to store and organize the memory of the cell. And lastly is the output gate, is used to controlling the flow of information out to the cell state. Next is chapter 3, research methodology. In this chapter will include the research framework, the research dataset, and the performance measurements. So the research framework, the research process can be partitioned into three main phases. So the first one is review on previous technique and data acquisitions. Phase 2, design and develop model. And the phase 3, evaluate and analysis the result. So the phase 1 is first collection of the research from article, journal, research paper, and biological database. So also review and study the previous deep learning algorithm, identify the problem background, and develop the research question. And lastly is verify research scope and the Python package. So the study and summarize the views and idea from the previous research have been done in this phase, so that the concept and the structure of the LSTM algorithm are studied to obtain a better view. And the dataset that have been used in this research is taken from the Stanford Open Vaccine. So the first objective, which is to analyze the mRNA vaccines and the existing computational algorithm and technique based on the LSTM methods can be achieved at the end of this phase. Phase 2 is design and develop model. So in this phase include the process of the exploratory data analysis, and this EDA is generally used to discover the hidden information and understand the data. So continue with the data preparation and the data augmentations, design and develop the algorithms, training and validate the model, and lastly is testing the model. So all of these details in phase 2 will be discussed in chapter 4, research design and implementation. So at the end of this phase 2, the second research objective will be achieved, which is to propose a LSTM method for the prediction of the reactivity for the mRNA sequence. And lastly is phase 3, evaluate and analyze the results. So in this phase, we will calculate and compare the MCRMSE value with the existing model. We also compare the MCRMSE result with other models, identify the limitation of model, and discuss on the result. So further discussion regarding the step and the result we obtain. So the last research objective is to validate and verify the performance of the LSTM method in terms of MCRMSE in the prediction of the reactivity of mRNA sequence can be achieved in this phase 3. So this is the examples of the features in the research datasets. Consists of the ID, which is a unique identification for each data. The sequence width consists of 4 nitrogenous bases, the structure and the predicted loop type, sequence score and the sequence length for the RNA. And this is the examples of the predicted features in the research, which are the reactivity and dose degradation rates. So continue to the part of the performance of measurement. So in this research, we use mean column-wise root mean square error. So as a method of FAIR, the MCRMSE have been applied to several topics in the Kaggle competitions. Ordinarily, the RMSE is only able to obtain single number of the evaluation metric for the prediction values. However, they consist of numerous requirements need to be predicted in this research. So therefore, the multiple different RMSE value will be generated for each column. So in order to simplify and make the data more understandable, the MCRMSE have been applied to calculate and get the average value along with the RMSE value for each predicted column. So that this is the examples of the equation for the MCRMSE. So let us continue to chapter 4, research design and implementation. In this chapter, we'll include the EDA process, data augmentations, and model constructions. So the first step in the EDA is to importing the Python's libraries. For example, Pandas library for manipulation of the data, namely for multidimensional error, Seaborn for visualize random distribution, Maplex for contribute in object-oriented application programming interface, and the TQDM for act as a progress bar that provides support for the nested loop. Next is loading to the dataset. So after import the Pandas package, the read function is applied to pass the particular URL where the dataset can be found in the Google Drive. So we use the read JSON file to read the training and testing dataset in Java format, and use the read CSV file to read the sample submission file in Excel. So next is explore to the dataset. So in this, we can see that the shape features is used to inspect the total amount of the observations and column in the dataset. It also used to identify the dimension of the data. So we can see that in the training dataset have 2,400 rows and 90 columns. So after that, we also use the isNow function to check the missing or null values in the data. So we also can see that there are no missing value in the training dataset. And this is the top 5 records in the training datasets. And this is the available 90 columns in training datasets. So same with the testing dataset, there are a total of 3,634 rows and 7 columns. And there are also no missing value in the testing dataset. So let's continue to the signal to noise distribution. And signal to noise is a measurement that compare the detection signal with the background noise. And the critical in the filtering data are the minimum values across the 5 condition must greater than negative 0.5, and the mean of the signal to noise across the 5 condition must greater to 1. If these both criteria are fulfilled, the SN filter is set as true. Other than that, will set as the false if both criteria does not fulfill. So this is the total sample of 2096, which the sample with signal to noise greater than 1, and total of 1,589 samples with SN filter equal to 1, and lastly the sample with signal to noise greater than 1 but SN filter equal to 0 have total of 509 samples in the dataset. Next is the base pairing probability metric. This metric is used to predict the secondary structure of the RNA molecules. Multiple alignments of RNA predict interaction between the RNA and measure the probabilities for each base pair in the sequence. So from this figure, we can see that the purple background indicates no interaction between the base pair, while the green color dot is identifying the interaction between the base pair. So continue with the data augmentations. So data augmentations is used to avoid the occurrence of the overfitting issue, while artificially increase the number of the dataset. So the effectiveness of the proposed model also can be enhanced through this augmenting the dataset. So the difference of the size between the training and testing set can be observed in this figure. So we can see that before augmentation in the training dataset is 2,400, and after augmentation is 4,800, while for before augmentation for the testing dataset is 3,634, while after augmentation becomes 7,268. So continue with the model construction. So this figure show that the model architecture for the proposed bidirectional LSTM model based on the BPPS data and the sequence data. So the model is first start with the feature engineering to extract the features and apply a sequence model to predict the mRNA sequence that responsible for the degradation by predict 5 reactivity value for every position in the sequence. So the features engineering will extract two types of features called numerical and categorical features. And first, the categorical features are encoded and embedding layers is adopted to capture the relationship in the sequence. Then the feature extraction is applied to extract the numerical features using statistical and mathematical equations. And finally, two types of features are concatenated to apply the bidirectional models. So first is the numerical features. So all of the following formulas is related on the pre-calculated base pair probabilities in the BPPS metric for each sequence. So every position in the sequence has a vector called BPP, which is the column in the matrix, and the n is the length of the BPP vector, and the BPPi's is indicate the probabilities of the given base with the position I for all the following formulas. So the sum and the maximum and normalized values for the vector probabilities have been calculated in this research. So continue with the categorical features. So since that the most machine learning techniques accept numerical features only, so pre-processing and preparing the data to convert the categorical features into the numerical features is necessary step for enabling the technique to process the data and extract the valuable information. So in our mRNA vaccine data, there are three types of sequence data to be encoded using the base method, which are the sequence, structural, and the predicted loop type. So first, the structural characters have been encoded from 0 to 2 from the open and close bracket and full stop character, and the sequence, which consists of ACUG character encoded from the number 3 to 6, and lastly is the predicted loop type, which encoded the character for BEHIMSX from number 7 to 13. So after encoding, the resulting code are fed into the embedding layer, which is a powerful feature that allows additional information to automatically insert into the neural network. So embedding layers are often used in the natural language processing. After data pre-processing step, encoding and embedding process are prepared and described just now in features engineering sections, and the features are ready to enter the sequence modeling as inputs. So in this research, the hidden layers in the proposed LSTM models at each layers consists of 259 in each direction, which means that the numbers of bidirectional layer unit is equal to 512. And this bidirectional layer is used to optimize the result, as the data can pass in the forward and backward directions to more flexible capture the information in the sequence data. So the global parameters that are used and configured in each bidirectional layer are summarized in this table. So we can see that we have 0.4 dropout values, 3 layers in the models, and sequence length of 107, and the predicted length in 68. So continued. In this figure, we can see that the summarize of the proposed LSTM model architecture. So we can see that it consists of one input layer, and the embedding layer, and the concatenate layer, the drop up layers, and the 3 layers of the proposed bidirectional LSTM model, and the tensor layer. So after undergoes this layer, we obtain this amount of total parameters, and there are no non-trainable parameters in this proposed model. So continue with the chapter 5, result analysis and discussion. In this chapter will consist the data augmentation evaluation, deep learning optimizer evaluation, and the comparative analysis. So the first one is the data augmentation evaluation. So based on these two figures, so the blue color indicate the MCRMSE value for the training dataset, while the orange colors of light indicate the MCRMSE values for the validation dataset. So next we can observe that the values of the MCRMSE is decreasing with the increasing number of epoch. So however, the MCRMSE values for the training dataset decline even more compared to the validation data and show a huge gap between them. So this means that this experiment has suffered the overfitting problem and the result getting from the proposed model might not be not so desired. So notwithstanding the result obtained from the augment data samples is more preferred compared to the data sample without augmentations. So this is because the average for MCMSE for the augmentations is more lower than the original data samples in this table. So next, continue to the deep learning optimizer evaluations. So in this section, the different optimizer are used to train the neural network. So the aim of applying the optimizer is to minimize the loss function in the machine learning algorithm by modifying the attributes such as the weight and the learning rate. Well, the way to change the parameter values or the running net is depends on the defined optimizers in the neural network. So different type of optimizer have their responsible and the method to reduce the losses and generate the most accurate outcomes. So therefore, the comparison between the stochastic gradient descent, root mean square propagations, and the Adam optimizer is conducted. And the result based on the MCRMSE values is shown in this table. So from this table, the neural network with SGD optimizer hold the highest average of the values among these three optimizers. So while the neural network with Adam optimizer present the lowest average of the MCR and SE values. Therefore, the Adam optimizer is outperformed compared to the HGD and the root mean square probabilities in this research. So lastly is the comparative analysis. So we can see that we compare the result between the proposed bidirectional LSTM in this research and with the hybrid model that proposed by the previous research with the similar dataset. So we can see that the MCR and SE value for the hybrid models is lower than the LSTM model with a difference by 0.101. So after that, the gap between the training and the testing set in the LSTM models is more wider compared to the hybrid model. So which means that the result that show hybrid model outperforms to predict the reactivity values of the mRNA sequence. So this might be caused by different type of machine learning algorithms implemented in the research. So although the research show that the LSTM is able to handle the vanishing gradient and the long-term dependency issues, however, the combination of the GRU and the LSTM proposed by the previous research show more effective. The GRU model incorporates two useful gates such as the update and the reset gates, so which allow it to perform better in the long sequence training samples. So this is because the GIU model does not have a memory unit, then it can just expose the whole hidden content without any control. So that might be cause the hybrid model outperform compared to the proposed bidirectional LSTM in this research. So lastly, it comes to the last chapter, chapter 6 conclusion, which include the achievement of the project objective and further work. So achievement of this project objective, we have successfully studied the mRNA-based vaccine and the existing computational algorithm for predict the reactivity of mRNA sequence in chapter 2 literature review. We also successful to propose a LSTM method to predict the mRNA sequence in chapter 4. And lastly, to validate the performance of LSTM method in terms of mean customized root mean square errors in chapter 5. So for the work, we'll go to study more on the structure and architecture of deep learning method to enhance the model performance, improve the existing method in terms of training and processing of the dataset, as lastly, validate and verify the performance of the proposed model in the prediction of reactivity for the mRNA sequence under various of conditions. So that's all my presentation. Thank you very much.